Cardiac muscle contraction is an intricate, precisely regulated process essential for the heart's role in pumping blood continuously. This process, known as cardiac excitation-contraction coupling, involves a series of well-coordinated steps, beginning with the initiation of an electrical signal and culminating in the powerful contraction and subsequent relaxation of cardiac muscles. Our journey begins in the heart's own command center, the sinoatrial node. Here, a small but mighty specialized cell, the pacemaker, is about to send out a spark, a tiny jolt of electricity, action potential. Once this action potential reaches a cardiac muscle cell, it causes a change in the cell's membrane potential. This shift in electrical charge opens up voltage-gated calcium channels, or the L-type calcium channels, on the cell membrane, allowing a small but crucial influx of calcium ions from the extracellular space into the cell. This initial calcium entry then triggers a phenomenon called calcium-induced calcium release. The small influx of calcium ions from the outside of the cell signals the cell's internal calcium reservoir, the sarcoplasmic reticulum, to release a much larger quantity of calcium through channels known as ryanodine receptors. As the calcium concentration rises within the cell, calcium ions bind to a protein called troponin C, which is located on the actin filaments in the muscle cell. This binding causes a structural change in the tropomyosin complex, which previously blocked the binding sites on the actin. With these binding sites now exposed, another protein called myosin can attach to actin and form cross bridges. The formation of these cross bridges is the start of cross-bridge cycle, a sequence of molecular events in which the myosin heads pull on the actin filaments, resulting in the shortening of the muscle cell. This action generates the force needed to produce the contraction of the cardiac muscle, which in turn enables the heart to pump blood out to the rest of the body. After contraction, the muscle must relax, preparing for the next cycle. To achieve this, the cell must reduce its calcium levels. This is accomplished by actively transporting calcium back to the sarcoplasmic reticulum using pumps known as sarcoplasmic reticulum calcium ATPase, or the SERCA pump. Additionally, sodium-calcium exchangers on the cell membrane helps remove calcium from the cell altogether. As calcium levels decrease, troponin releases calcium, allowing the tropomyosin complex to return to its original position and block the actin binding sites once again, ending the contraction and enabling the muscle to relax.